Drugs are chemical or biological substances that cause a specific physiological effect. Medical providers prescribe them to treat an enormous variety of physical and mental health conditions. All drugs are built to act on a specific target within the body. Often these targets are proteins, but as our understanding of genetics increases, new drugs targeting specific regions of DNA or RNA are becoming more prevalent. There are thousands upon thousands of compounds that might prove useful, but how do researchers decide which substances might treat which conditions? Once they've narrowed down the pool, how do they know if a treatment is working? And how do they decide whether a treatment is safe? In the past, many drugs were discovered by accident, but today the process is far more organized. Usually, new treatment strategies are developed in response to a new and better understanding of what causes a condition or disease. When they understand what causes it, how the disease progresses, what body systems are affected, and what, if any, treatments have been tried, researchers can begin to design a new drug. Using computer modeling, today's researchers often begin by creating a detailed digital map of the target associated with the disease. Usually, the target is a specific protein in the body. Then, they digitally reverse engineer a molecule, the prospective drug, which will bind to the target. The prospective drug must be complementary to the target protein. That is, it must be the right shape, size, and charge in order to attach properly. Once researchers have designed a molecule they believe will bind to the target, they need to build the molecule in a lab. Depending on the size and complexity of the substance, this can be an extremely time-consuming and inefficient process. Chemical reactions involved in drug manufacturing often create a large amount of waste in return for only a tiny amount of usable product. If the needed molecule is very large and has a complex shape, such as with insulin or human growth hormone, creating it via a series of chemical reactions is far too inefficient to be useful. Instead, researchers have learned to harness genetically modified bacteria to build the needed molecules, which are then extracted for use in patients. Even when the researchers have successfully created a product they believe will bind to the intended target, the process is far from over. The multi-step FDA drug development process is meant to test for efficacy and for safety. Drugs are evaluated thoroughly, and those that don't measure up are not allowed to proceed to the next phase. The new drug must pass through preclinical trials, several phases of clinical trials on humans, and a thorough review by the FDA before it can begin to be manufactured at scale and then go to market. This process often takes years. Despite the rigorous testing and approval process, it's impossible to replicate every scenario in a clinical trial. Even after a drug is in use, researchers and doctors monitor a drug's performance very closely. They continue to collect data and record any instances of adverse reactions, unforeseen interactions with certain other medications, or any other safety-related information that might be useful to prescribers. Drug development is a long and complex and expensive process if it's to be done correctly and safely. Only a small fraction of prospective drugs are safe and effective enough to be brought to market. In future, as our understanding of biomolecules and genetics increases, hopefully we will be able to better predict which molecules will be successful and further streamline the design process.